So at the end of the day, all of these different options of receptors, of signaling molecules, of transduction pathways is there to get us a response, okay, or an output response, right? Signaling molecule would be your input, the response would be the output. And those responses are something that we're trying to regulate in our cells, okay? In some cases, it may be a single response we're trying to regulate. Other times, I can use one signaling molecule with a handful of transduction pathways to regulate multiple cellular activities. It all depends on the pathway that you're dealing with. But regardless, those responses tend to fall into a couple of different categories. If it's a response that ends up happening in our cytoplasm, it's usually a way to regulate some sort of enzyme, and a lot of those tend to be metabolic pathways. If I'm using my response as something that's going on in my nucleus, then I'm probably regulating the actual creation of enzymes. So here I'm regulating their activity, how much are they working, how little are they working. Here I'm regulating whether or not they get produced, or maybe it's not an enzyme, maybe it's some other sort of protein. All right, so that's where the idea of them working as transcription factors and regulating gene expression comes in. In some cases, that cellular response might be something physically about the cell itself, so something like cell shape. All right, so when we get to talking a little bit about plant hormones, the regulation of those hormones is to help move plants, because they can't get up and just walk away, to help them move around and do certain things to help because of external stimuli. Okay, and so some of that I'll come back up there as well. So here's just an example of regulating enzyme activity. All right, we're using epinephrine or adrenaline. That goes through a host of activation of different protein kinases. All right, here's your secondary messenger as cyclic AMP, and that's going to end up regulating the conversion of glucagon, which is stored glucose in your cells, into normal glucose, so that we're probably using this in something like a fight or flight idea. All right, I need all that glucose in my blood because I need the energy to either run away from my predator or fight somebody who's trying to come and take over my territory, whatever the case may be. I need that pathway to get triggered. Here's one where I'm regulating the actual production of an enzyme or protein synthesis. So again, here's something like a growth factor. So remember, those are utilized as a way to signal cell division. Okay, and that growth factor is going to come in, it's going to hit a receptor, also going to go through a phosphorylation cascade, so kinases being phosphorylated down a cascade, and that's going to end up in my nucleus taking an inactive transcription factor and activating it. That active transcription factor now binds to my regulatory sequences in my DNA, turning on a gene, allowing RNA polymerase to better bind to that promoter sequence. Off we go with transcription, which ultimately gets me my messenger RNA, and then I go through translation to get that protein, whatever it happens to be. Now, just like I want to turn these pathways on, I've got to have ways to turn them off. And the termination of a lot of these signaling pathways is really pretty simplistic. I attach the signaling molecule, I start up the pathway. If I take the signaling molecule out of the receptor goes back to its inactive state, and I shut down the pathway. So sort of like putting the key in your ignition, turning the key, you turn your car on. As soon as you take the key out, you've turned the car off. All right, and so that's a lot of, really simplistically, how a lot of this works.
Hi, it's Mr. Anderson, and welcome to Biology Essentials, video number 38. This is on signal transduction pathways. Signal transduction pathways are very important in the actions of cells, but they're sometimes misunderstood. So I wanted to start with an analogy. And so when Jimi Hendrix plays the guitar, he would vibrate the strings on the guitar. Those would then be transduced. In other words, the pickup in a guitar, the electric pickup in a guitar, has magnets and wires inside it. And what it does is it transduces that message of the wires into an electrical signal. It then goes into an amplifier where you can make it really, really loud. And so we can hear it. And that's what he was famous for. And so signal transduction pathways in cells do the same thing. It starts with a message, and that message is in the form of a chemical message, and then that's transduced into actions within the cell, and it also can be amplified. And so signal transduction pathways all work in the same way, and there are a couple of ways that their actions work. Sometimes they'll actually modify a protein or change the shape or the conformation of a protein, but mostly what they'll do is there'll be what's called a phosphorylation cascade. In other words, a phosphate group, which remember carries energy, will be passed off from one chemical to another to another to another until it eventually has an action. And protein kinases are important in that. And so the example that I'm going to give you today, a message comes in, a message is picked up by a receptor, and so this line we could say is the cell membrane on the outside of the cell. That message will dock with the receptor, and when it does that, in this case, it'll change the shape of that receptor. The example I'll give you is what's called the G protein receptor. Then we'll have transduction. Transduction, remember, is when we're switching that message and we're changing that from a signal message on the outside of the cell to a message within the cell. We use what are called secondary messengers. The one that I'll give you an example of is called cyclic AMP. It's a very common messenger in cells. And then that'll eventually target the cells. In this case, it's going to target cells in the liver, and it's going to make them release glucose from glycogen. And so that's kind of a signal transduction pathway. It starts with a message, and then it eventually has some kind of a target within the cell. And so let's get started. This is an animation of this typical liver cell and how epinephrine can affect it. And so we're going to go through, and I'll pause at a couple of different spots and explain it. So epinephrine is the messenger. Epinephrine is going to be given off from the adrenal gland. It's going to move throughout your body, but it's going to especially affect cells in the liver. So epinephrine is going to dock with this receptor. And in this case, it's called a G protein receptor. And so the epinephrine is what's called a ligand. And so a ligand is going to be a chemical. It's a chemical that can't make its way through this cell membrane. It can't make it through this hydrophobic region, so it's going to dock with the G protein on the outside. G protein is a protein that's embedded within, it's a snaky looking kind of a protein. It's embedded within that cell membrane. So it's got a portion on the top and it's got a portion on the bottom. It's got these units on the bottom. They're called subunits. And so what happens is when that ligand attaches, in this case epinephrine, with the G protein, it causes a conformational change in that protein. So it's changing the shape of the protein. And what happens when it changes that shape is it actually releases one of those alpha subunits. And so one of the subunits, called the alpha subunit, will be released, and it's going to move to a protein just right down the way in the cell membrane. This protein is called adenylyl cyclase, and it'll make sense why it's called cyclase in just a second. But essentially what it is before the actual alpha subunit comes, it's an inactivated enzyme. In other words, it's an enzyme that's not working yet. It hasn't changed its shape, so it's actually a functioning enzyme. But once the alpha subunit is in place, it's ready to do its job as an enzyme. And in this case, what it does is it converts ATP, adenosine triphosphate, into cyclic AMP, or we sometimes call this CAMP. Now, what is ATP? We know that has 3 phosphates attached on the outside. It carries energy mostly in cells. We're very familiar with how ATP can be converted to ADP when it drops off one of those phosphates, but it can also drop off 2 phosphates, and that's what happens in this case. So now it becomes AMP, or monophosphate, but there's also a cyclic portion to it, and so it adds right where we come off of the sugar. It's actually going to make a cyclic portion. I'll put a picture in here of that molecule, ATP, and now we've created these messengers. Messengers are going to spread throughout the cell, and this is called cyclic AMP, and those secondary messengers in this case are going to target something called the protein kinase. Protein kinase, it's made up of a number of different subunits of protein, but kinase means it does something or it does action. In this case, this protein kinase is going to have two catalytic subunits. Catalytic means things that are going to speed or speed up chemical reactions. And then it has these two regulatory subunits. And so as long as the regulatory portions are attached to the catalytic portions, protein kinase is inactivated. It's not going to do anything. But let's watch what happens to the cyclic AMP. It'll actually bind to those regulatory portions of the protein kinase, and it releases the catalytic portions. And so now we have this cascade. In other words, we have this cascade of energy. Those catalytic portions are going to become phosphorylated. In other words, they're going to pick up energy from ATP, and they're going to become activated. And they change now from a green to kind of a yellow activated color. They then can act on enzymes within the cell. Sometimes they'll act through a number of different cells, molecules within the cell. In this case, it's going to drop off that phosphate to phosphorylase, and it's going to activate phosphorylase so it can release glucose from glycogen within the cell. Now, once we don't have that ligand attached anymore, we don't make that cyclic AMP, then the whole thing is going to shut down again. And so the signal transduction pathway is simply a way that we can take this message and we can move it throughout the cell and then have desired consequences within the cell. Okay, so let's do a little review. And if you've ever watched the show Dora the Explorer, there will be times where she just kind of pauses and looks awkwardly at the person who's watching the show. And so that's where you have to jump in and help a little bit. So let's do a little bit of review. So we start with this at the top. We've got epinephrine at the top. And so what do we call this? That's right. It's called the ligand. A ligand is a chemical that can't make entry into the cell, but it's going to attach to the receptor. So this is called the receptor. Do you remember what that's called? That's right. It's called the G protein. And so what will happen is that ligand will attach to the G protein. It's got a number of different subunits. This one right here on the end is called the alpha subunit. That's right. Hey, if you're not getting any of these, you may want to go back and watch the earlier portions of the video. We've got this over here, and this is probably the one that you're going to struggle with the most. What's the name of that? Oh, that's right, adenylyl cyclase. And so what happens is the alpha subunit is going to attach to adenylyl cyclase. We then have an enzyme that's functioning. It's going to take in these little starbursts. Those aren't starbursts. They're called, that's right, ATP. ATP will then be converted to cyclic AMP or CAMP. And cyclic AMP are now going to go work on this protein kinase. That's right, protein kinase. It has two portions that are catalytic and two that are regulatory. That's right. Okay, so the cyclic AMP is going to move over to that. But let's watch what happens for a second. Because there's not just a few cyclic AMPs in a cell. There are going to be lots of cyclic AMPs and lots of protein kinases in the cell. And so remember when we talked about our analogy of Jimi Hendrix, this is where we can amplify the message. So we just have this one ligand up here, but we can have all this action going on. I didn't want to animate all of that, so I went back to just one protein kinase. So we will free up the catalytic portions. We now add energy to them. What's that called? Phosphorylation. That's right. We're going to add a phosphate group to them. They're going to turn yellow. And now they're able to pass on that phosphate group to phosphorylase. So it's activated, and it can break down glycogen into glucose within the cell. And so that's the signal transduction pathway. It's fairly simple. It's got a lot of steps in it, but it's just like playing the electric guitar. And so I hope that's helpful. Hi, it's Mr. Anderson, and welcome to Biology Essentials video 39. This is how changes in signal transduction pathways can have huge effects. I'm going to talk about poisons and diseases, but I wanted to start with one of the most dangerous animals that we have on our planet. It's the California newt. California newt, you could pick it up, you could hold it, it's no big deal, but if you were to eat it, you would die just within a few hours. And that's because the California newt produces a toxin on its skin. We used to think the newt produced it, but we now know that it's a symbiotic bacteria that's producing this toxin. It's called tetrodotoxin. It's one of the nastiest neurotoxins that we've ever found. It affects the sodium ion channels in the nerves, and so what it does is shuts them down, our nerves can't work correctly, so those cells don't work, and then we can't breathe and we die as a result of that. Tetrodotoxin is found in lots of other animals. So pufferfish, if you're familiar with that, and in Japan, hundreds of people each die just from pufferfish poisoning each year. Or some of the cephalopods, like some of the really toxic octopi, also have this tetrodotoxin inside it and shuts them down. It also goes by another name, and that's called voodoo powder or zombie powder. If you want to Google tetrodotoxin, you'll find the story of a Haitian zombie that was created through voodoo processes. And I don't really believe, no, I do believe in zombies, and they're coming, so watch out for that. But it's kind of an interesting story of how they took this one person, fed him some tetrodotoxin poison, and supposedly kept him as a zombie for years. Interesting story. True? Who knows? So let's start with signal transduction pathways. So again, if you don't know what that is, make sure you watch the video on that. Essentially what happens is a messenger will dock, a ligand will dock with a receptor. It transduces that message, it sends it to a number of secondary messages, and then it targets parts of the cell. So it has an action within the cell. So it's a great way to send a message into the cell and also amplify that message. Now, signal transduction pathways can have disruptions. So whenever you have a series of events, you can also have a series of unfortunate events. In other words, we can have blockage along this way, and that can have huge consequences. And so the two cases I'll talk about is anthrax poisoning and how that affects a protein in a specific signal transduction pathway, and then one you're maybe a little bit more familiar with, and that is diabetes mellitus, or regular just diabetes. So let's start with anthrax poisoning. Anthrax was in the headlines years ago, post 9/11, and what was happening was somebody was sending anthrax spores. Anthrax spores are essentially bacteria that have dried up. They just have their genetic material inside there, and then they're just waiting. So if you breathe in anthrax, it goes into your lungs, and then this bacteria starts to grow. So this is Bacillus anthracus. Will start to grow. It feeds on your lungs, and it produces a toxin. And that toxin can disrupt cells and eventually can cause death as a result of that. And so how does that work? How does that affect us? Well, when I talked about the signal transduction pathways, the one that you're probably familiar with was how a ligand can bind to the G protein. That G protein can eventually release a alpha subunit, which then makes cyclic AMP. Cyclic AMP is a secondary message. We can amplify a message, and then we can target parts within the cell. Now, what we've started to realize is that the poison found within anthrax actually targets this enzyme, adenylate or adenylyl cyclase. And so it changes the shape of this. So it changes the shape of this protein. It now can convert ATP into cyclic AMP. And so that is the way that anthrax poison actually works. It's just blocking a part of the signal transduction pathway, and then it has huge consequences just in the cells in which it affects. Another one you're maybe more familiar with is diabetes. Diabetes uses a signal transduction pathway, not diabetes, but there is a signal transduction pathway inside all the cells in our body. So we have an insulin receptor. An insulin receptor is going to sit on the surface of your cells, and when insulin docks with that, it has a series of events that go on within the cell. So what are some of the events? Well, it's going to affect this glucose transporter. So I like the name of that. That's just called GLUT. And GLUT is going to allow a cell to take in glucose, make use of it, store it as glycogen, or make use of it inside the cell. And so if you have type 1 diabetes, what's going on there? Well, if you have type 1 diabetes, you're not producing insulin. You're not producing insulin, therefore it can't dock with that insulin receptor. And so that's why you have to get insulin shots. Here would be a pump working. And that's essentially producing insulin within the body so we can actually set up the signal transduction pathway. If you have type 2 diabetes is actually affecting the insulin. You're still making insulin inside your body, but that insulin receptor is ignoring that message coming from the insulin. And so that's going to disrupt the signal transduction pathway. We can't activate the GLUT. We can't take in the glucose as well. And so again, signal transduction pathways are awesome. It's a great way that we can transduce a message. We can amplify that message, but it's also a great place to target as far as diseases and poisons and toxins. And so I hope that's helpful.